Bismillahirrahmanirrahim. So now, when the secondary oocyte is released from the ovary, as shown here, it is released as a secondary oocyte, excuse me, and it is released into the peritoneal cavity over here, right here. And this is the fimbriated end of the fallopian tube, and there are cilia on the inside of this tube that are beating. And because of the beating of those cilia, a suction current is produced, which is going to suck this ovum that has been released from the ovary into the peritoneal cavity, and it is sucked into the uterine tube, fallopian tube, where if it encounters the sperm over here, it is fertilized, and if it's fertilized, it completes the second meiotic division. Remember that it was arrested in the metaphase of the second meiotic division, and if it is fertilized, it gives off the second polar body, and the nucleus of the sperm will fuse with the nucleus of the ovum. The nucleus of the sperm is 23N, 23 chromosomes, haploid, single N, and 23 chromosomes in the ovum with a haploid number, and when both of them fuse, they restore the normal number of chromosomes, which is 46-N, and a zygote is formed. This happens on day 1 of fertilization. So if due to some reason, some abnormality, the ovum cannot enter the fallopian tube and it remains inside the peritoneal cavity, and 6 days they pass, so the implantation will occur inside the peritoneal cavity, and the cells around the zygote are going to erode, and they're going to erode the peritoneal surface, and they're going to implant into the peritoneal cavity, and development will begin inside the peritoneal cavity, and that's very dangerous. It's a ectopic peritoneal pregnancy, and the danger is that as the fetus will grow and it will expand, more and more erosion will take place, and when the blood vessels in the peritoneal cavity they get eroded, they will bleed, and since there are no uterine muscle fibers, normally the implantation occurs in the uterus as shown over here. If there are no uterine smooth muscles of the uterus to contract and stop that bleeding, there is a danger that the female can bleed to death in this condition. So that's a clinical correlate. So that's why we have to be very careful about those pregnancies. We have to terminate them if the implantation occurs inside the peritoneal cavity, or implantation occurs in the tube, same thing happens. So what happens now that fertilization has occurred on day 1, and the cilia keep beating and they keep pushing this ovum slowly and steadily, and the goal is to push this ovum from the fallopian tube into the cavity of the uterus. But as it goes along, it keeps dividing and developing. So on day 2 it becomes a 2-cell blastula, on day 3 it becomes a 4-cell blastula, on day 4 it keeps on going that way until day 4. This is the 64 or the 32 cell stage, and it's called a morula. It's a solid ball of cells. But remember that this type of division is a cleavage division. A cleavage division in which simply 2 cells become 4 cells. There is no growth in between cells. Normal mitosis, when the cells they divide, after division they grow and they increase in size. There is increase in the size of the cytoplasm and the organelles, but over here there is no such thing. It's just simply cleavage means cutting into smaller and smaller pieces. Two cells become four cells. One cell is half the size of the original cells, and the four cells become eight cells, and one cell is now 1/8 of the original, the size of the original cell. So it's just cutting the cytoplasm into smaller and smaller pieces until you reach this stage, or a solid ball of cells, and this is known as the morula at day 4. Once it reaches day 4, now on day 5, uh, something different starts to happen. And instead of having a solid ball of cells, now there is a fluid-filled cavity inside this ball, and this is known as the blastocyst. And there is differentiation of cells. One of the cell group of cells is the outer cell mass, is known as the outer cell mass over here, and the other group is the inner cell mass. The inner cell mass is known as the embryoblast, and these are the cells from which the future embryo is going to develop. It forms the embryo. The outer cell mass is known as the trophoblast, and the troph means nutrition. So these are the cells that are going to provide nutrition to this embryo on day 5. And there is a fluid-filled cavity inside this uh, uh, zygote at this time, and that's why it is known as the blastocyst. So on day 6, the blastocyst reaches the uterine cavity, and once it reaches the uterine cavity, now this is the enlargement over here, the trophoblast, they start developing, they start differentiating into two types of trophoblasts. These are the cells which are known as the cytotrophoblasts, and these are the cells which are known as the syncytiotrophoblasts. In syncytium, the difference between the cytotrophoblasts and the syncytiotrophoblasts is that the syncytiotrophoblasts have no cell membranes in between their nuclei. So it is the it is one huge cell which is multinucleated. It has many nuclei. And the cytotrophoblasts, they start dividing and they start producing syncytiotrophoblasts, and the syncytiotrophoblasts start eroding the surface of the endometrium and the embryo in order to facilitate implantation of the embryo. So this happens at the end of the first week. So we've gone from ovulation to fertilization to cleavage division, and then to the formation of the blastocyst. And at the end of the first week of the 6th day, the implantation occurs, and then the development starts to occur uh, when the embryo gets implanted inside the cavity, inside the wall of the uterus.